So before I jump in, I just really want to highlight that this work that I'm going to talk about today represents efforts from a lot of people and a lot of hard work, and it's a real honor to be here today to show you what we've been up to down in New Zealand. So Enterococcus faecium, or E. faecium, typically it's harmless. It kind of lives in all of our guts. The problem is when it gets into a hospital environment, particularly where there's vulnerable people, it can cause infections, things like urinary tract infections, bloodstream infections, and so on. And then, obviously, in the hospitals, there's an antibiotic selection pressure, which can trigger these bugs becoming resistant to last resort antibiotics. In this case that I'm going to talk about today, vancomycin. So in New Zealand, we are in a very fortunate situation. So here I've just kind of highlighted all of the vancomycin-resistant Enterococcus faecium that we've tracked over time. So these are all clinical cases. So every time a diagnostic lab confirms vancomycin-resistant E. faecium, it gets recorded. So the general trend is it's quite low, but we can see the cases are slowly increasing, and this would put a burden on an already stretched healthcare system. Now, I say New Zealand is in a very fortunate situation because our friends across the ditch in Australia, they only monitor VRE in bloodstream infections, and you can see the difference between all of our clinical cases and just the bloodstream infections in Australia. So New Zealand is in this really great position to be able to do something a little different. Don't think it's working. There you go. So today I'm going to be talking about vancomycin-susceptible Enterococcus faecium and why we decided to track it. So not all E. faecium is resistant to vancomycin just yet. And there's a couple of mechanisms on how this bug can pick up this resistance. It's mainly through horizontal gene transfer and mobile genetic elements. And the mechanism I'm going to focus on today is this transposable element named TN1549. So how this works is if you can imagine this piece of DNA circularizes and it floats around the environment, and when it comes into contact with an AT-rich region in a bacterial DNA, it undergoes some DNA rearrangement and strand exchange. So if you think of a bubble landing on the surface of water and then the bubble kind of disappearing into the water, that's the situation that's happening here. And if you need to know more about this, then I have highlighted this publication that came out in 2018 in Cell. So this is where we are in the world. The map on the left shows New Zealand. We comprise of two main islands, the North Island and the South Island. And then the purple that I've highlighted is the Wellington region, which is where this study has taken place. And now the map on the left just shows the district health boards that we're focusing on in this study. So Capital Coast and Hutt Valley District Health Board. And I've overlaid the population density per square kilometre. And then the yellow dots just show where our public hospitals are. The lowest yellow dot being Wellington Hospital. This is where our on-site sequencing is based. The red dot is where ESR is. That's who I work for. And we act as a reference lab. So we collate a lot of data from all over the country to really bring in that together. The dot next to ESR is Kiniperu Hospital. And then that dot on its own, that is the Hutt Valley Hospital. Now, the way outbreak response works in New Zealand, so we got the micro labs, they do their thing, a lot of work, process a lot of samples. When they suspect there's an outbreak, they take a sample from the patient, do their diagnostics and their phenotypic testing. And if it's a pathogen that's on a list of things, then they would submit a slope culture to ESR. Now, only certain isolates are required to be sent to ESR for surveillance. So when we receive them, they get cultured. The Nososomal Infections Laboratory will then send those isolates for sequencing, where they'll report back multi-locus sequence typing and any clustering analysis back to the decision makers. Now, what we're trying to do, we're not trying to replace this business as usual process. It's well established, but we've put a MinION sequencing device in the hospital so that when they're growing these bugs up and doing their phenotypic testing, we can scrape a bit off the plate, jam it through a nanopore, and get a species ID and an MLST that bit quicker back to the decision makers so then they can implement their infection prevention control response. So we started this in January 2022. So between January 22 and December 24, we had 265 clinical specimens. 208 we were able to successfully generate an faecium genome. And as you can imagine, most of them were from Wellington Hospital. That's where our on-site sequencing is based. We do have a couple from Hutt Hospital, and then it's just an ad hoc basis where Kinniprue Hospital, or we get some samples from the community as well. And the vast majority of them, 196 of them are vancomycin-susceptible. Like I said, not everything is required to be sent to ESR. So the VRE, we did have a couple of cases. So any time a VRE is detected, it gets sent to ESR. So this is a very broad clustering analysis. So it's based on core genome SNPs. So what I've done here is downloaded some publicly available assembled faecium genomes. They're represented in gray. I've included the ESR VRE genomes. They're indicated in black. And then the Wellington genomes that we've sequenced with Nanopore only. And done this clustering analysis. And one thing that stuck out to us straight away is we had this cluster pop up that was spread between Wellington Hospital and Hutt Hospital. There's a lot of patient transfers going on. So we wanted to investigate this further to see what was going on. So now we get into the realm of the high-resolution SNP analyses. So we generated a high-quality reference genome representative of this lineage. We used that as a reference to map the nanopore reads and to call the SNPs. And then we built a phylogeny. And then we used some temporal analyses and a tool called BEAST, which is a very complicated mathematical formula that puts a really lovely timeline on the x-axis that you can see. And the way we can use this is we can see how these strains are related to one another and when their common ancestors have emerged. So, for example, the 2020 dot on the far left, that would be the last common ancestor to this lineage. So it existed around about 2020. And another great thing that we can do about this is we can show, so this lineage was comprised of two main clades, clade 1 and clade 2. Clade 2 differed from clade 1 by 2 SNPs in a COP-A gene and a LYS-C gene. These genes could be involved with virulence, but we won't know until some phenotypic testing is done on that, but, handy information to know. I've just condensed down the 30 other genomes because the point I really want to highlight is in purple. So we had one patient. So we've been aware of this lineage since August 2023 from tracking. And in October 24, we had a patient come in with a lung infection. Sample was taken, vancomycin-susceptible faecium. 54 days later, after vancomycin treatment, took a screening sample from the same patient. This differed by just one core genome SNP, but it did acquire that transposable element that I mentioned earlier on. So against my colleague Letitia as well, she prefers seeing the data, but I tried to simplify this for the audience. So the top strand is the patient vancomycin-susceptible infection, so that first sample. And then the second strand is the screening sample taken 54 days later. So we can see where exactly in the genome, so that AT-rich region, that this transposable element popped in to that second isolate that was collected later on. And now this is just a BLAST-N comparison, so just comparing the nucleotides of that transposon. The top one being our patient screening sample, so the VRE. And we can show that it's very similar to a transposon that's been found on a plasmid from the same species. It's also been found in a different species, Enterococcus faecalis, and also Clostridioides difficile. So this is quite valuable because it's likely a reservoir of this transposon but we need more data to be able to confirm that. And like I mentioned at the start of the talk, there was a lot of effort that went into this, so I want to take a minute just to thank all of the various teams at ESR, in particular the Antibiotic Reference Laboratory, and then I want to name Max Bloomfield, my clinical partner, who's a bit of a unicorn because he has a lot of enthusiasm for genomics, and Jesse Martin down in Dunedin, who is a microbiology lab scientist and has found his new love in genomics and has done a lot of work building up his bioinformatic expertise. And with that, I just want to say thank you.